That's a really good one.
All right, let's go ahead. Get started. All right, so I sort of will admit that I kind of changed my mind on how I wanted to cover this kind of last minute. So we'll see. But so what I sort of decided is that we should focus just on the receptors first, because there's a lot of them for mechanosensation, and then sort of looping in audition and vestibular a little bit, because that's really mechanosensation. They use their own class of receptors as well. And yeah, so we're going to focus on that. And I wanted to start by asking a question that might not seem relevant, but hopefully we'll get there. This is based on, a lot of times I realize class is really influenced by whatever I'm listening to or reading, and this is definitely an example of that. So if anybody's ever read the book Packing for Mars by Mary Roach, no? Good, that'll make this seem like a, no, I'm sorry, this has nothing to do with this. I came up with this myself, so there. So why are astronauts recommended the scheduled times to pee, or to use the bathroom in space, right? Why don't you think about it for a second and then talk to the people around you, with the hint that mechanosensation is something there, right?
I'm sorry. Yeah.
All right, does anybody have anything? Questions? How do we know when to pee? That's, I guess, the bigger question, right?
So are there like mechanoreceptors of some sort, but they're like on the bottom of your bladder? So when you're in space, the gravity is there, or like on some pressure, like maybe some of the pressure is not there to like trigger those mechanoreceptors or something?
Yeah, that's pretty much it. Is that yes, there are mechanoreceptors in your bladder, right? And with the more sensing, they would sense general stretch, but more so they're primed because we evolved in gravity, and so you would expect pressure to be exerted, you know, predominantly towards the bottom, right, where gravity is. And apparently, this was news to me when I learned about it. Like a lot of astronauts never will have an urge to pee because of this. That can be a really bad problem, because eventually if your bladder fills up so much, it actually like pinch itself off, I guess. So then you can't pee, right? So they actually have to schedule this in because their the system is not working correctly. So there's, I think, a sort of a use case for mechanotransduction that maybe we don't think about all the time. All right. So I had also mentioned, yeah.
Do you think that when they come back to Earth, where like the gravity, are they more sensitive?
I never thought about that. That'd be kind of terrible.
Like,
I can imagine the post-flight news conference of just like people like getting up all the time. Like I have no idea if it makes sense to me to think that might be true. That as your body would adjust, because that's the general commonality of sort of like sensation in general, as you adjust to sort of a new baseline, that that when you know you would be in this more extreme situation again, that they would be, but that is not something I have heard of. So it's kind of interesting. All right, so The other thing, we've sort of touched on mechanoreceptors a little bit in that when I talked about the Nobel Prize, that was largely what it was given up for this year. It was for at least the people who discovered some of the different classes. And I thought a kind of interesting way to start would be to kind of go over one of those papers that discovered them. This paper is actually fairly recent. We're just going to go over two figures of it. Where it's from 2010. So in the realm of Nobel Prizes, that's a pretty quick turnaround. I feel super quick. Where they discovered the channels for touch, right? And so here's what it's showing. The first thing, they basically started to do a screen, right, where they don't really know exactly what it is, but they want to try and figure out which of the genes is responsible for touch. And what they're showing in figure 1 here is basically they have this cell line, right? It's actually a neural cancer cell line, which the reason for using that is it's actually kind of hard to grow neurons, right? They don't divide usually. So it's really hard to get them. And this happened to be a human cell line, so even harder. But because it was a cancer cell line, they divide and it's human, that solves some problems. And what they happen to show, and they're trying to show you, is that it happens that this particular cell line is actually, the cells themselves are directly sensitive to touch. And so what they're showing here is that if they appear, this is we've seen voltage clamps and such things that sort of look similar like this. But what they're actually showing is that they're patching a cell here, and then they're just kind of pushing on it in a defined way, right? And the more that they indent the cell, this is the scale bar is actually microns here. So the greater they make an indentation, the greater the current that they see flowing through, right? Indicating channels in the open by sort of just pushing on it, right? The other main point of this figure 1, sort of defining it, is that they do a different type of patch, right? Remember, we could do either excise patch or patches where you could look at single receptors. That's what they're doing here. And they can show that they can find single receptors, right, that are activated by touch, which is very important for sort of setting up their system. That they have narrowed down in this one cell line, there seems to be one receptor / ion channel that can sense touch, right? So then they use this to do a screen, right? And right, so that is the basis of the next part. I thought the way, all right, oh yeah, so the take home is that these two cells, the N2A cells, have a mechanically activated ion channel. And they know that, and they're going to discover it. And really, that discovery comes in this next figure I'm going to hand out here. So this figure has really, I'm going to say, 3 important parts. And I want you to sort of read through. So I had a professor once who said, if a figure is well written, you should be able to drop it with the caption, the figure legend, and figure out what it means from there. So we're going to try this. See if this is well written enough to do this. All right, so take a little bit and read through this, and then what I'm going to ask is we'll break in, I'll have different groups do it, but eventually we're gonna have 3 groups explain the figures. So, different parts of it, but just take some time for us to read through. Oh, 2 points I brought up here that you might not get sort of reasonably so. They use the term MA, that means mechanically activated or mechanically activated current. So, and then they talk about siRNA. siRNA is a way to knock down a gene, right, or inactivate a particular gene, fairly specifically, right? So I'm hoping people at least made a pass through that by now. So now it's gonna break into groups, and we're gonna go over this by panels. So I'll assign 2 groups to each, and I'm only gonna call on one to present. That's the only way I can figure out to make this work. But if you guys will turn around and work with them, you can cover A, you guys can do B, and then C and D combined across the back, and then there's only 2 of you as well. You still get A, B, and then C. All right, C and D together, right? First off, does anybody have any questions to ask at this point?
So how can you tell which color is blue?
So I don't think it's actually it's actually important to know which color is which, but I mean, I have the color figures up here, right? That's why I like blue, the rest are red, right? And then in this one, it's slightly more important, but not really.
Oh, okay.
Is that
Can
enough?
you show A for a quick second?
That's
Okay, that's red and that's blue and the rest of it, okay,
Yes.
thank you.
They reference it a lot. I did debate this, but it doesn't actually help you interpret. I don't feel like once. Okay, any other questions? All right. Give you a few minutes. You should be able to try and tell us what they did and what they found. That's the key here.
Yeah, yeah, it's all the schematic currents, like, of like, to solve this. And no wonder. Well, the different, I was reading like the different types. Yeah. So like each one shows that this shows how to solve. Well, it's not. Yeah, it's like different. No. Oh, this is in a random. Well, it's moving. What do you mean by? So it's all the same. Yes. Like the one.
So.
Oh.
Like the mayonnaise.
Yeah.
Okay. Because it says it's a way to inactivate the plants and directly off and it makes
So-
the letters.
But it's a control.
That was.
And then change that. It wasn't.
They're treating them to a. They're not treating us. They're using us to treat.
Okay, so the safety effects as
On. There's only two. I know. So the last line of the average. So the black line is just average. So the black line in the middle of those is like the average overall. And then the dash lines are like plus or minus.
Yeah, so it's like the.
Yeah, I shouldn't say. I don't know what that means anymore.
How are you guys doing? Okay. How about over here? Good. All right. No? Okay. You want to take a shot?
No, I see it.
So maybe the more really, what's the one?
Those are all the different.
Right. Yep.
But I don't know what the candidate is.
It's a G. Yeah, that's what it is. What is the difference between the scramble? So scramble is really, so what you do to make a scrambled one is you have to have a specific sequence for an siRNA. And that should specifically just not them. But it could be, maybe that's the letters has some off target. So what you do is you create a randomized version, right? Same letters, random order, and you also transfect that in to prove that, oh, actually, the scrambled version you have to deal with that. And that's sort of controlling for all the other. Only due to that specific sequence, it should be specifically not. So what are they doing in this? You know, so there's a little picture in the upper left, sort of the same picture. So they've got one patch by patch, so they're measuring, in this case, current, that's what your axis is. And the other one is focus. So What's it? And the other key thing is that the x-axis is water, so the effect is sort of minimum. All right. Anybody need, I think it sounds like we've moved on mostly. All right, so we got 2A groups. I'm gonna admit, you guys have 5 people in the group, so I was gonna choose you. Somebody want to explain panel A here?
So we were super positive when you got into like some of the scrambled stuff, and please correct me if I say anything that we did not say. But essentially we're looking at the average amplitude of the N2A cells depending on what they've been transfected with. Is that-
Yes.
Okay,
Yep.
cool. So we're on the right track. The black line in the middle, well, I guess it might be kind of blue on the screen, but in the
No,
middle
I
of that
think
like hot mess.
The middle one is still gray. The
Okay,
black-
so that's technically like the overall average.
Yeah,
The red dot is gonna be the, like, what's it called, like the mutant siRNA. And the blue dot is scrambled, was our understanding.
yeah.
We weren't totally clear on why there were so many other data points. That was confusing. We didn't know if that was like way, like a lot more N2A cells, and each of those represented a different N2A cell just for the sake of having a larger N, or if we were missing some other variable.
So y-axis here, which is what each one of those different dots is a different one, is candidate number. What did you have an interpretation of that? They aren't super clear on what they mean by that.
Skipped over that. Not gonna lie. Well, because it says list of candidates available in table F1. Yeah, I didn't know it was.
Right. But what do you think that might mean? Just interpreting the word candidate. Right.
I would assume they were looking at different N2A cells to determine which would be the best for this kind of analysis or something like that.
That's a reasonable, yes. I sort of explained it to you guys. So can you explain it?
We didn't have it in the paragraph, but we sort of guessed that those were different genes that they were looking at to try and figure out how this mechanoreceptor works.
Right. So they know that these cells from the previous figure have a mechanosensor, and these different candidates are different genes they're going after using the siRNA to knock them down. So they test, I thought it was like, the scale doesn't make sense because I think somewhere else they say 60 some, but whatever. They test approximately 70 different candidate siRNAs, right? And all of them they're going through, they all land within the sort of standard deviation of whatever it is expected, except for that one, right? This candidate's different. That candidate, I think they give it the name right there, piezo1, right? Yeah. So they found one that's way different. I think, so what's the actual experiment they were doing here? Did you get that?
I mean, I was just trying to like isolate an outlier like that.
Yeah, so what were they measuring? What were they, what's the sort of,
Amplitude. So,
so the x-axis, the big A there is actually meaning amps. So they're measuring currents, picoamps is the P, right? So what they're doing, it's actually showing up here, is the experiment I talked about earlier, where they've got one patch pipette that's measuring current, and another just glass pipette that's sort of poking cells, right? And so they poke it a certain amount, and they see how much current they get through at the sort of peak. And a key thing here is this is actually a logarithmic scale. So on average, you're getting, this is probably something like 300 picoamps, and this guy is only doing about 8, right? So that's a huge difference, right? So if you want an experiment, and this is something I want to talk about at the end, this, which I think in some ways seems pretty simple, but like once you understand what's happening, like that's a Nobel Prize, right? That experiment was the one, right? This is the one that figured out. This guy turns out to be really important for a lot of things. So, yeah. All right. So I think that explains that one pretty well. B, let's go. I'm going to alternate to this side for B.
So this is kind of the same method as A with the voltage clamp, but on the one that kind of poking the cells. But it's showing the data, we were thinking more like condensed into the average for each gene. And so the kind of key point that we latched onto is that the scrambled siRNA has a much higher average response than all the other piezo-1 siRNAs.
Right, so that should be, I hope, they give you nicely colored so that like it really draws your attention to that fact. But the scramble is a control. And since I explained it to like half of everyone explained it, right? So what they did is you have an siRNA is a specific sequence, ideally if it's working right, that should target a gene and basically shut it down so you don't make that gene anymore, right? The RNA, it actually targets the RNA and destroys it, right? However, you want to make sure you're not just having some random off-target effect and you're just making like the cell sick or something. So what you do is you take your sequence that you initially used and you randomize all the letters in there and you transfect that in as well. That's your scrambling, right? You scramble up all the letters. And if everything's going well, you should see no effect, your control versus an effect. And then what they did is they actually went sort of another step here. The siRNA 1, 2, and 3, they made 3 different siRNA sequences, all targeted at the same piezo gene to really nail down the point that yes, they are. There's a big history. This is why it's sort of interesting that siRNAs at this time were pretty like hot technology, right? Shortly afterwards, we found out they're not super specific, and we got CRISPR, and now hardly ever do they get used anymore. But they are still useful, and they were useful for screens like this, right? The pools are where they sort of use multiple different siRNAs all against targeting it's the same thing, right? And that's fine. What is did anybody read what the number above means? I use this throughout a little bit.
The number of cells tested.
Yep, right. So different conditions, they tested different numbers of cells, right? That's somebody patching each one, in a cell, measuring, right? I think that's it. All right, so C and D, let's go back to this side.
So what we're looking at with C is the induced current affected by negative pressure. So it's scrambled out of the selected current. There's no insensitivity suppression of
Yeah, I think in a lot of ways, this is sort of more, this ends up being a really important point to make, right? So they want to sort of nail it down in every way possible. And this is just showing you a little more detailed information about what different pressures elicit these currents and the sort of detailed current traces. Yeah. All right. So if we go back to this figure, right? As I said, pretty straightforward at what they're doing. This was a really important question in science. Why did it take us until 2010 to answer it if we just had to do this? Anybody have this? In some ways, I don't expect you to answer it, but I'm sort of curious, like, why didn't we know this already? It seems like something we ought to know. So the key here is really that this siRNA technique they said isn't interesting was only a few years old at the time. And that was what allowed us to screen through it. Because before this, if you wanted to delete a gene in a cell, each cell was like a couple month project. Now you could do all of these in like, a few weeks. And that was actually a previous Nobel Prize. That was only about a decade before this, right? Because I think that's kind of an interesting example of how this all builds upon each other. All right, so that's how you discover the receptor. I thought this figure was kind of interesting. This is showing, so they discover piezo-1 using this technique. Then they used the bioinformatic approach, not so different from what you guys were doing in lab the other day. They took the sequence for piezo-1, they blasted it, and they found, oh, there's something else that looks pretty close. Probably, let's call that piezo-2. It ends up being actually maybe even more important for mechanosensation. And we'll talk about that in a bit. But you can see here they made their little evolutionary tree of that. What I think is really interesting about this is a super old gene. You look at it. This is like chordata, so like vertebrates, right? It's in invertebrates. It's also in bacteria. It's in mold. It's in plants, right? When we know this now, like if plants have a response if they sort of get crushed, right? That is partially mediated by piezo channels that are in those plants. So this idea of needing to sense sort of pressure changes or mechanical force is all super old and related back. They also did this other experiment to show where those channels are expressed, right? And that's what's basically being shown here. They use a RT-PCR method to see piezo 1 and 2 in these different tissues, right? So hopefully we can answer this now. Why did they look, if you look at, this is all the different tissues they looked at, right? Why did they look in the bladder? This is meant to be a softball. We've kind of talked. Yeah, Sarah?
Like what we talked about earlier, like, there's some kind of receptor. I don't know if they knew at this point there were mechanoreceptors in the bladder, but maybe there's some kind of receptor.
So they did know there was mechanoreceptors. They knew that all sorts of tissues were sensitive. We had no idea what the receptor was, right? So I looked for it to find it. Why did they look for these channels in the DRGs right here?
What is DRGs again?
That's what I was hoping you remember. But if we don't, that's okay, right? Yeah. And what do they do? Yeah,
It's
sure.
like, it's like in spinal cord, right? It's something either like sensory, receiving, or like input.
Right. So spinal cord, this is one thing I do always memorize myself. Dorsal, usually sensation, ventral, usually motor, right?
Oh,
So,
yeah.
so these are the sensory cells, particularly of touch, right? And they find it there and very high levels. So why do we have these channels? If DRGs are whatever is important for touch, right? Why do we also have piezo channels in your skin cells? But we're gonna come back to that in a second. That's more of it, right? And then last, compare these two graphs. What do you think? What are the most interesting differences between the two graphs? Take a minute and look, and then you can talk to the people next to you. All right, so what do we see as key differences between where the two channels are expressed? Jane?
The big difference between ERT expression between the two, I mean, it's almost polar opposite.
Right, yep. And from this figure, the piezo 2 will actually get labeled the neuronal piezo, and the piezo 1 will say it's a non-neuronal tissue. So I think it's a little bit not quite as clear. So any other big ones you brought up?
Increase.
Yeah, so I think that's the other big one. And I think this is how they get there. Skin is up high and it's pretty low there. And the other ones there are some differences, but not in the same ballpark as those two. And also, I'll just bring up the point, there's also massive expression of both of them in the lungs. Yep, I think that should make sense. Yeah.
So then for the, you said that piezo one tends to be categorized as non-neuronal. And then piezo two as neuronal, does that mean like the non-neuronal is more like stimulation based, whereas the neuronal is more communication based? Like they're the between cells that communicate to other cells, I guess.
I don't think that's a completely crazy thing to say, but I'm not sure. That might be an overgeneralization
Okay.
as well. Yeah. I think that makes sense in some capacity. Yeah. All right. So this picture is actually from your book. This is the structure then of the piezo channels, and it's kind of a crazy thing, and very hard to work with for this one. But if you look, they're showing. Remember, we've talked a lot about G protein coupled receptors. Those are 7 transmembrane receptors. They go zigzag through about 7 times. Those are considered big membrane receptors, right? Well, this one has, it's so big we can't even know for sure exactly how it does it. This is guessing 38 transmembrane domains, which it zigzags through the membrane in that. And then here, if you look at sort of, this is actually an EM topography model that was made of the actual structure of the channel. And the white is sort of where they think lipids fill in. But anyways, the sort of green and yellow are the real channel. And I think the top-down view is a little more indicative of what it is, where it's got these, it sort of almost looks like a little hurricane in some ways, right? And then the center here is the actual channel that opens in response to mechanostimulation, right? And so based on what you're looking at here, right? How does this sense force? Right? Can you imagine how that channel in the center might open? Why don't you talk to people next to you for a minute? Think about, so this is a channel in the membrane. Think about what happens to a cell when you squish it, when you push it, right? Does anybody have any thoughts?
Which one are we looking at? Are we looking at like B or A?
I mean, both could be of use. This is like a cartoon diagram, and this is literally what it is, right? This is the sort of side view of how this actually folds in the membrane. You can sort of imagine the membrane going through here. This is a top-down view.
What's the squiggly bits between the,
Which squiggly thing?
like up and down?
Yeah, so this is all one protein sequence, and it's just zigzagging through the membrane. Yeah. if you have a balloon, right, and you sort of push your finger into it, what happens? It's pressure, right? At some point, it becomes harder and harder to sort of push your finger into it, right? Why is that? What's providing that resistance? I suppose that's sort of true, but it's not the answer that is most relevant to my question. Yeah,
I mean, if we're comparing it to a cell, like the balloon itself would be like plasma membrane. And the stuff inside of it would be the cytoplasmic area. So there's stuff in there that prevents it from like moving farther in. And when you push it in, it increases the pressure because like there's, I don't know. And it depends on how stretchy it is too.
so something has to stretch, right? Tim, did you have something?
Yeah, I was just gonna say that like, the more you push on the surface, the more tension that surface has.
Right, yeah, right. You can't blow a balloon up forever because that sort of the rubber of the balloon is stretching, it only stretches so far. So when you push a cell, you're stretching that plasma membrane, right? And that plasma membrane is sort of all in here. And so if it's being stretched, right, you can almost imagine how these are sort of looking like little hooks in that membrane, and they're sort of being pulled open, and that physically pulls open the channel that's in the center here, right? I think that's, are we on board with that? I always think that's a pretty cool thing about how that physically works, that you can sort of see how that might. Right. All right, so how does this work in the skin? Here, so this is showing example of like, there's a lot of different skin receptors, and because this is, I'm going through this a bit slower than I intended, I'm not sure we're going to get to all of them today, and that's fine. But a sort of generic type of skin receptor for touch is this slow epinephrine here, right? So you have specialized skin cells, right? They have piezo, like we said, and they give signals to this neuron itself that also has the other type of piezo channels. They're actually both able to sense this stretch and force to a degree, right? And so we can see here, we can measure them. This is showing Merkel cells, right? That's the type of cells here that can sense the pressure. And depending upon the sort of force they're given, they start firing action potentials in response to that, because opening these piezo channels lets in a whole bunch of different currents, right? One interesting thing, if you looked at the graph, it actually says it's not in skeletal muscle. Well, the reason they looked in skeletal muscle, and it actually turns out to be wrong in the long term, is because we have lots of stretch sensors that sort of wrap around our muscle fibers, and these are responsible for proprioception. People know what proprioception is. I always like this, if you put your arm behind your back and bend it, you know you bent it, right? How? You can see it, right? That's proprioception. We know where our limbs are in space, predominantly because we have all sorts of stretch sensors that are wrapped around our muscle fibers, they're wrapped around our tendons, they're measuring the length of all our joints as we move. It's very important for us to have these, right? And this is the piezo receptors are what are sensing that through different channel setups like this. So other Nobel Prize was really run for these, the trip channels that are important for temperature. Actually, both of the guys kind of contributed to this. David Julius is predominantly, he's the one who discovered these capsaicin sensitive TRPV channels. They both discovered simultaneously these menthol ones that are cool. But right, so these channels, what shows on here is they respond to different temperatures within a range, right? They're not super specific, but the fact that we can sort of add up how responsive all these are gives us a pretty specific range, especially when we're outside of our comfort zone. In this sort of area where we are okay, sort of right around room temperature, we actually don't have very good discrimination between different temperatures. But as you get sort of more at the extremes, we're more sensitive to, right? this figure is always interesting. So if you look at it, they're showing you that, they respond to temperatures, that's what's shown here. But they also each have, or most of them have different chemicals, right? I actually didn't have this one. The one for cold here responds to things and some chemical in garlic and radish, and I forget what it is. This is menthol from mint. This is camphor oil, which also is one I didn't know for that one. I'm familiar with that. And this is capsaicin from hot peppers. Another key point that right here is that we tend to classify the ones more normal temperature as just thermosensors. And then the ones that get at the more extremes are nociceptor or temperature sensitive nociceptors, where nociceptor means pain sensors, right? The response you will get from your brain from these ones at the extreme is much less about temperature and much more about pain. Right? So, and this might be a philosophical question. I think it's not. So are trip channels mechanically gated or chemically gated channels? Why don't you talk to the people around you for a second? Right. So would you consider these chemically gated or mechanically gated or something else? I guess there's always that. Everybody too close to the end of class for such a question. Shane.
We say chemical, and we were just thinking because like they react, like with like the pepper, for example, or it like I think the pepper is like touching like your sensitivity and it's like the spice and the pepper is what we were thinking. And so it's like the chemical and the pepper that makes the spice is what reacting with.
Great.
I know what the conclusion is.
Okay. I think I buy that. Right. What do you think these receptors were? Why did we evolve them in the first place? Why did we evolve to have these receptors expressed and work this way? Do you think that we had a strong evolutionary need to sense the spiciness of foods? Or how much mint is it? Yeah.
I would say we evolved this to understand like the standard not necessarily maintain spaces, right?
So temperature is probably the key here, right? We evolved them for temperature and sort of the battle of evolution. I would say we as in humans that evolved them, it was something much earlier, right, that evolved these because of all sorts of and then this is sort of an example of counter evolution, right? I mean, the main story we think we know the best anyways is capsaicin, right? So why do peppers have capsaicin, right? It's actually slightly neurotoxic. We don't tend to like it. It's a noxious chemical, right? So we don't eat them in their seeds, right? And therefore they get spread more easily. In particular, birds don't have TRPV1, right? So they eat all the peppers they want, and they actually will pass through them, and they will spread their seeds, and that's how peppers are naturally thought to spread, right? So that was sort of the answer. All right. So in general, we kind of talked about one of these. There's sort of two ways the sensors, receptors can sense force, right? We talked about the one that sort of stretch, where they're sensing the changes in the membrane tension. And the second idea is that you can have sort of links to something else that sort of pulls them open, a different protein or something. And that second case seems to be what happens in sort of the vestibular and auditory system. And I think I got enough time to go through the basics of this, right? So this is a vestibular neuron. It actually looks a lot like the same hair cells that we have in our ear for sound, and that they all have this sort of base-like bottom, and then these thin hair cells on top. And what I'll show in a little bit when we talk about auditory is that these hair structures on top are all sort of connected to each other, right? And when they're pushed one way or the other, they can open or close channels by sort of pulling against them, right? So in your vestibular system, and we'll come back to this more a little bit later, but I just want to sort of intro it, right? You have the structure, your whole inner ear, this part that looks like a snail, that's for your auditory system. But this part here that is hard to draw because it's very much three-dimensional is called your semicircular canals. And you have three circles, so one that goes up, one that goes sideways, and what does it go? And then this way, right? So you get all three, you can sort of detect three dimensions, right? And that's what's showing here. And within these tubes, right, there's fluid, endolymph, right, that flows around there. And there's also some little crystal-like otocysts, right, that sort of float around. And this flow is how we sense our movement, right, particularly within our head, but it's how we sense acceleration and position of our head. So as we're going faster, right, it's going to cause flow to move sort of backwards, depending on which way. Or if you're falling, it's going to go upwards, right? If you turn your head one way or the other, this is what it's showing here. One side, the hair cells get deflected one way, and the other side the other way. And your brain can interpret all of that pretty quickly to say how you're moving, right? This is also why, though, it really can only sense acceleration, right? Because it has to cause flow. If you are moving at a constant high speed, we don't sense movement really, not through this system, right? Only visually can we sense it. And that's why you can get in sort of weird circumstances based on that, right? Quickly here, this shows a little better. These are the same looking cells, very similar hair cells in your auditory system that are going to detect the vibrations that are sound. And what I like about this figure is a couple of things. First, in the lab, we can literally sort of take something and sort of push on these, right? And measure currents. And that's what they're showing here is you sort of push one way, you get the current moving, you let back, current stop, push, right? And you can see how a current moving like this and creating a wave is much like a sound wave, right? And we can detect them here. The other thing, I hope you can see it, but you can literally visualize these in this DM. You can sort of see them here, but these like different hairs, right? There's a tip link right here. Now, hopefully you can see a little hair-like structure that connects the bigger hair-like structures, I guess. And that is, the thought is, I would say it's a thought, we pretty much know it's true, but we don't know the details, is that you see here, one tip link is connected here, and then it's connected to another channel. So when they're pushed, this pulls them open that channel, and it lets some ions in. And that's going to lead to finer exponential, right? What I would say, though, is this is kind of the last one in our sensory system, right? We don't know 100% accuracy what that channel is. A lot of people looking for that channel and have been for decades. It's one of these things where I almost feel like we won't know ever confidently because there's probably been a dozen papers where somebody's published, here's the channel for hearing, and then somebody comes back and does a different expansion and says, those people are complete idiots, right? And it goes back and forth like that. Interesting though, I think we all assume, or a lot of people assume, we would have found this because mutations, we have a lot of genetic evidence, right? We should have found a mutation in that channel that would cause deafness, and we basically know all mutations that cause deafness, and we haven't found it that way. So that's confusing. Either that channel is so ubiquitous, right, that if you have a mutation in that channel, you have much bigger problems than just hearing loss, or there's something else that we just can't explain. But interestingly enough, I think I could say almost all mutations that cause deafness are in the proteins that make this, right? That seems to be the real key structure for here. All right. So this will be an okay place to end. If we, yeah, I think we have enough time to sort of, right? So let's think about space again. We've got talked about a lot of different mechanoreceptors. When we started to go to space, right? We set animals up first because basically scientists and doctors sort of on land were like, we have no idea, right? Like, are people just going to explode when they go into space because they can't handle the pressure change or changing gravity? A lot of people thought you would go blind. All sorts of senses were shut down. Your heart would pump so hard you would explode. Nobody had any clue, right? And so it turns out it was actually pretty okay. There's some things that happen, but nothing nearly as what we predicted. So let's think about this for a second, though. It isn't, we are affected by the loss of gravity. We talked about it with the bladder earlier, but what do you think is potentially most affected out of sort of the ones we talked about? Which of your mechanosensory symptoms would be disrupted by a loss of gravity? This is not all inclusive, but I thought this is what I wanted to sort of, right? Should talk to the people next.
It makes sense, but I don't really like the. Your body's
All right, does anybody have, what's your best guess?
Oh, it's similar, just because the gravity situation, you don't necessarily need your legs or your arms to, you don't stand up straight, so if you're up there for long periods of time, your muscles start to contract.
Yeah, so yes, I think vestibular is the one I understand, at least having never been in space, that is actually most messed up. And I think the reason that they give actually is more, everybody talked about those otoliths that sort of float around in there. They're held in place by gravity. So without gravity, they just kind of float all willy-nilly through your semicircular canals. And when you hear astronauts talk about it, it basically, it takes a couple weeks and somewhat variable. They give them lots of drugs for this, but like extreme disorientation, right? Because there is no up or down anymore in your world ever, right? And even so, like we send students on this, the what they call the vomit comet, the thing that simulates microgravity by going up and down. This is why that's called that, right? Is your vestibular system doesn't care for that, right? You're turning gravity on and off throughout the time, and the thought is generally that when your vestibular system becomes out of line with the other things, it thinks you have ingested something terrible, and it tries to get rid of it, right? Yeah.
Is this why people get like motion sickness, is because their like vestibular system is messed up between that flow?
Yes. Well, I mean,
Visual.
motion sickness is mostly due to conflicting senses, right? Where your vision's telling you one thing and your vestibular system is telling you something else, and your body's like, pump the brakes here. That's bad news, right? Yeah. All right. Anybody else say something? I think there's a few other we could say, but I think vestibular is probably the big one. We do notice, I think, for baroreceptors, that's a big one. I mean, we talked about bladder would fall under there. Blood pressure can be a little bit wonky, but nothing terrible. Eye pressure, similarly, is like that. So some effects on that. But most of the other senses generally unaffected for what we know. All right, so we will end there. Let me get. Right, so I've realized I told thesis this on Monday, but I'm gone Friday. So we don't have class Friday. This was also why I realized I left you guys in the dark on a few things. Like we said, I mean, it does say this on the syllabus that we had an exam that should be due. But in my mind, and I changed it on all my calendars, I knew I was gone and I messed it up and I pushed it back a week. Also, SFN has changed, so I don't really have to take any time up with that. So regardless, that's why this is all, I think it's a change that everybody's okay with, that both not have class Friday and that the exam is pushed. So hopefully that's not messing anybody up. I didn't give you a reading assignment in the end because it felt like not enough time and then we were gonna have this extended time. You will have a reading experiment and either a quiz or some sort of questions for Monday, right? And mainly what I'm covering here is from a chapter that I think I will not actually have you read, in the hopes that I can cover it in class without that. All right? All right, so I will see you guys Monday, then, all right?